Hello dear student, today in this lecture I am going to discuss about the structure of HIV. Now you know that HIV actually is a virus that causes AIDS. HIV means human immune deficiency virus. In this lecture we will discuss about the basic structure of HIV, which is enveloped, matrix, capsid, genome, and in genome there are enzymes, reverse transcriptase, integrase, protease, and we will also discuss the GAG gene, pol gene, NVIV gene, and accessory gene of HIV. So let us start that what is actually HIV. HIV is a retrovirus. What does retro mean? That actually this virus or these viruses are having enzyme reverse transcriptase, and this enzyme do reverse transcription. What does it mean? It means that actually these viruses convert their RNA to DNA. This process is known as reverse transcription. Now normally transcription is actually the formation of RNA from DNA, but these viruses, they convert the RNA into DNA. This process of conversion of RNA to DNA is called reverse transcription. So these retroviruses, they are called retroviruses because these viruses are having reverse transcriptase enzyme. Now HIV, there are two types of HIV, HIV1 and HIV2. HIV1 having subgroup, they are called lentivirus. So first of all, we are going to discuss that what is the basic structure of HIV. The basic structure of HIV consists of the following parts. First part is enveloped. Actually, envelope is a lipid bilayer that HIV gets it from the host cell membrane during budding or during the infection. When HIV infected the a cell, now this HIV bud from the cell and gets it enveloped from the cell membrane of the host. Now this envelope is having specific glycoprotein. One is mushroom like. Look at this. This is mushroom like, and this mushroom like glycoprotein is known as GP120. It is envelope protein. So GP120 is mushroom like, and the function of GP120 is actually it has receptor, CD4 receptor on the host cell, and it help in the attachment. Another transmembrane. Look at this. These transmembrane glycoprotein, these transmembrane glycoprotein are called GP41 glycoprotein 41. It help in the secondary attachment. So HIV is actually envelope virus, and this envelope is having special type of glycoprotein. One is called GP20 and the other is called GP41, and these two proteins help in the attachment of HIV to the CD4 positive cell or T helper cell. The next structure of HIV is matrix. So look at this figure. These are actually this structure, this rounded structure below the matrix. This overall structure is known as matrix. and this matrix is made of specialized protein. This protein is called P17 protein. It forms the matrix. The third part of HIV is actually capsid, nucleocapsid, and you should note for MCQs that the nucleocapsid of HIV is rectangular. Look at this structure is actually rectangular, and this is called nucleocapsid. Now the question arises that which type of protein form the nucleocapsid of the HIV? This protein is called P24. Now we are going to discuss the genome of HIV. Actually, the genome of HIV consists of two single-stranded RNA. One RNA is this, and the other RNA is this. These two RNA are present in the genome of HIV, and these RNA are actually positive polarity. They show positive polarity. Positive RNAs are those RNAs which directly acts as a messenger RNA. So HIV is having two RNAs, so they are said to be diploid. Beside the RNAs, HIV genome also contains the three very important enzymes. One enzyme, they are associated with these RNA. One enzyme is known as reverse transcriptase. Look at this RNA, and this RNA is having enzyme associated known as reverse transcriptase. Now what these reverse transcriptase do? When HIV attack on the cell and infected the cell, after entry, this reverse transcriptase actually synthesize DNA by using the genome RNA as a template. ये HIV का ये क्या करता है कि ये जब सेल को इन्फेक्ट करता है तो रिवर्स ट्रांसक्रिप्टेज HIV की RNA से DNA बनाता है. उस प्रोसेस को हम रिवर्स ट्रांसक्रिप्शन कहते हैं, और इस वजह से HIV को हम रेट्रोवायरसेस कहते हैं. Another type of enzyme is known as integrase. Integrase integrate the viral DNA into the cellular DNA. Actually, after the formation of DNA, this DNA are inserted into host DNA. The enzyme responsible for integration is integrase. Another enzyme is protease. What does protease do? Actually, protease cleave the various viral precursor protein. When virus form small protein, a large protein inside the host, these large protein are further cleaved. They are cleaved and form various proteins such as GP120, P17. They are the actually function is performed by protease enzyme. Now, if we talk about the structure gene found in all the retroviruses, so now the question arises that what gene are responsible for these protein? One gene is called GAG gene. Actually, GAG gene form P24, P7. Actually, these protein form the nucleic capsid. Now, matrix is formed from P17, and this P17 protein is formed from GAG gene. This is formed from GAG gene. Now, another is actually pol gene. Actually, pol mean that these pol gene form three type of enzyme: reverse transcriptase, that is protease, and that is integrase. And we discuss the function of the reverse transcriptase, that it transcribe RNA genome to DNA, and protease cleave the precursor polypeptide, and integrase integrate the viral DNA into the host. So GAG gene is responsible for the formation of nucleic capsid protein, and pol gene is responsible for these three enzymes. Another gene that is known as actually NVU gene. NVU gene form the enveloped glycoprotein, that is GP120 and GP41. We have discussed actually these two spikes in the envelope, and actually the transmembrane is called, the transmembrane glycoprotein is GP41, and the mushroom like is known as GP120. Now what is the function of GP120? GP120 help in the attachment to the CD4 protein. Now what is CD4? Actually CD4 are the receptors that are present on the which type of cell? T helper cell. They are T helper cell. They are present on T helper cell. They are also present in macrophages. And actually the virus when come, they attach to the T helper cell. Why this T helper cell has CD4? They are CD4 positive cell. They are CD4 positive cell. And actually this GP120 they attach, GP120 attach to the CD4, and the secondary attachment is done by which type of glycoprotein? They are GP41. GP41. Now we are talking about the HIV also have regulatory gene. Regulatory gene means that they regulate the viral replication in the body. HIV is regulatory gene, that is one is called TAT and the other is called REV gene. Actually regulatory gene found in the immune deficiency virus that are required for replication. Actually TAT means that it activation transcription of viral gene. It activates the transcription of viral gene. And the other is REV gene. Actually these REV gene transport of late messenger RNA from nucleus to cytoplasm. It means that when the messenger RNA are formed, in the nucleus, actually this messenger RNA formed in the nucleus, and this RNA actually they are transported to the cytoplasm. They are transported to the cytoplasm by which protein? By REV. Actually they are produced by REV. Now a regulatory gene found in human deficiency virus that are not required for replication, they are called accessory gene. Accessory gene are those genes that are not required for replication but they are required for the pathogenesis. One of these gene is called NEF. Actually NEF decreases the CD4 protein and class 1 MHC protein on the surface of infected cell, induces death of uninfected cytotoxic T cell, important for the pathogenesis. Now you know that normally what do MHC molecule, the function of MHC molecule is to represent the virus to the which type of cell? To the phagocytic cell or the T cell. But this NEP protein actually they class switch, they reduce the MHC molecule, so they are not represented to the immune cells, so the immune cell cannot recognize the HIV and so it multiply. Another is WEP gene. WEP gene is actually it enhance the infectivity by binding to ApoBig3. WEP bind to ApoBig3 and actually an enzyme that causes hypermutation in retroviral DNA. Now VPR gene transport viral core from cytoplasm to the non-dividing cells. You should note it. And another important is VPU. VPU actually enhance viral release from the cell. When a virus, complete virus is formed inside the cell, so this virus is transported to the other cell by the help of VPU. In the next lecture, we will discuss the replication and pathogenesis of HIV, and also we will discuss the treatment, and also we will discuss that why vaccine are not formed against HIV virus.